Thank you. And hi, my name is Siqi Shen, and the topic I want to talk about today is this scGAD, or single-cell gene associating domain scores for exploratory analysis for single-cell Hi-C data. And this scGAD allows for the integration between single-cell Hi-C and other SCs. Nearly 2 meters of the genomic material for the human being are packed into chromatin in a micrometer scale nucleus, while remaining accessible for various biological functions. The organization of the chromatin is thus extremely relevant to the biological function. Information about the interaction frequency among all pairs of loci on the genome. The data structure is like something shown on the right of this slide, which is similar to an adjacency matrix of a graph. Each row or column of this matrix is the specific bin or a specific location on the DNA strand, and each entry in this matrix is the interaction between two bins. The birth of single-cell Hi-C allows for studying the genome-wide chromatin interaction at an unprecedented resolution. And shown here are 4 Hi-C contact maps. The first row are 2 bulk Hi-C matrices. They're from the same cell line, but different studies. And even if they're from the different studies, visually speaking, they're very similar. And the second row are single-cell Hi-C matrices from same cell type and same studies. And due to the different sequencing depth and the biological variation, these two matrices looks completely different. And because of this variation and because of this more complex structure of the single-cell Hi-C comparing to assays like single-cell RNA-seq, which is basically just a cell-by-gene matrix, current methods for single-cell Hi-C analysis is very scarce. I listed some major methods shown here, and most of them are just doing normalization, imputation, or clustering using single-cell Hi-C solely. And no method at all is trying to do integration between single-cell Hi-C and other assays. The area of integration is growing rapidly. So basically, there are 2 directions. And the first direction is experimental, which scientists are trying to measure different biological features from the same cell. And in this direction, there are only 2 papers that tries to measure methylation and the chromatin organization from the same cell. And the second direction is computational, where scientists are trying to merge and project cells from different studies and, of course, different assays into the same space. The figure shown on the right is from the second paper shown here, and it represents the spirit of this kind of approach. And again, no computational method is developed for integrating between single-cell Hi-C and other assays. And we talk a lot about how hot it is for the area of integration, but we didn't talk about why we need integration, and especially why we need this for single-cell Hi-C. So listed here are three reasons, and the first 2 reasons are better cell type annotation and better cell type separation will be what we will be talking about this in the next few slides when we look at the data set. The third reason is more about biological, but unfortunately, due to the scope of this talk, I won't talk about it here, but it's also very important. And with that, we can start to discuss a little bit about the data set we'll be using. The first one is the Tan et al. 2021 dataset. It uses neonatal mouse brain and measures single-cell RNA-seq and single-cell Hi-C respectively. That means they're not measuring these 2 biological features from the same cell. And if we look at these 2 cell types in single-cell Hi-C, they are like neonatal neuron 1 and neonatal neuron 2, and they are vaguely defined cell types. So this raised the the problem for this data set, that those subcell types are hard to annotate just based on single-cell Hi-C. And this is also the reason I talked about in the previous slide why integration is important. The second data set is Li et al. 2019, which measures human brain, and it measures methylation and scHi-C simultaneously from the same cell. Shown here are the clustering methods for scHi-C data alone, and the regions highlighted here are those neuronal subcell types. And it is easy to see what the problem is in here, that subcell types are hard to separate based on scHi-C data. And this corresponds to the second reason why integration is important. And with these problems and with the data set, we can get into our method introduction. Our method is called scGAD. It is a very simple method. The first step is to merge or sum all the interactions inside the gene body. And then we can get a cell by gene matrix. After getting this matrix, we first eliminate the sequencing depth effect by dividing by the depth value of each cell. And then for each gene, we subtract its mean and divided by its standard deviation, so that each gene has a mean 0 and a standard deviation of 1. And this allows us to remove all the explicit or implicit gene level biases. And with the method in hand, we can start an initial analysis. So first, right now we have two data sets of scGAD and single-cell RNA-seq, and both of them are cell by gene matrices. And what we do is that we take the average for all the cells in each cell type, so that we can get two cell type by gene matrices. And they have the same dimensions since we're using the same number of genes. And what we do is that for each cell type, we take the two vectors and we directly visualize them like these scatter plots shown here. And we also calculate the correlation between these two axes. And if the correlation is high, it means there are high correspondence between single-cell GAD and single-cell RNA-seq, and the integration is possible. And indeed, it is. Like all the correlation are larger than 0.5. And we also do this for another data set, and most of the correlation are also larger than 0.5. And this proves the relationship between scGAD and single-cell RNA-seq. Next, we take all those cell type specific marker genes from single-cell RNA-seq. We look at their scGAD value. We take the average of those scGAD values for their corresponding cell type, and we directly visualize them using a heat map. So shown here, there's a diagonal pattern. And what this means is that the cell type specific marker genes defined by single-cell RNA-seq are also cell type specific for scGAD. And this further proves the relationship between scGAD and single-cell RNA-seq. So after all these, we can get into the exciting part of projection. So the first data set is this Tan et al. 2021, and we're going to project it onto single-cell RNA-seq data from the same paper. And remember, the problem for this single-cell Hi-C data is that there are sub-cell types that are hard to annotate. And this is the projection result, since it is very hard to see like whether these two axes are really overlapping. We also have a data source plot along with the two UMAP plots for these two axes. And from the data source plot, we can see that there is a perfect overlap between cell types between these two axes, and it means the projection is a success. And if we focus on the dash square shown here, these the neonatal neuron 1 and neonatal neuron 2 are appears in here. What also appears in here are these cortical pyramidal cell, interneuron, median spiny neuron, and the hippocampal pyramidal cells. And if we make use of these cell types from single-cell RNA-seq, we can find or annotate these two cell types without being confused about what they really are. And with the same cell type, we also want to see if the projection is successful using other assays rather than only single-cell RNA-seq. And PairTag is a perfect example, a perfect data set here, since it like simultaneously measures gene expression and the virus prompting states. First, since they also have a single-cell RNA-seq, we just want to do the projection to see if it is consistent if the single-cell RNA-seq matrix is changed. And the projection is success. the cell types are still overlapping perfectly with each other. And if we look at the neonatal neuron 1 and 2 in here, we can also annotate them with the single-cell RNA-seq cell types shown in here. And next, we will project our scGAD onto this H3K27AC. The projection is a success. And also with the H3K4MB1, and the projection is also a success. And this means this kind of scGAD will be successfully projected with all sorts of assays. And for the projection part, finally, we have another data set, Li et al. 2019, and we will project it onto the brain initiative data that is single-cell RNA-seq. And remember, the problem for this Hi-C data set is that sub-cell type can be hard to separate only based on single-cell Hi-C data. And the left plot is the previous, is shown previously that the like statistical methods trying to cluster the cell types using only single-cell Hi-C. And the right part is the result of projection. And all those neuronal subcell types are in these red boxes. And as you can see, these cell types are successfully and almost perfectly separated from each other. And this means that as long as there is an appropriate gene expression data or other kinds of assays, the projection will be a success. And since there are ample data available, I'm pretty confident that for most of the single-cell Hi-C data available, we can find one or even more like those data that can lead to a successful projection. And finally, since we all know sequencing depth has an important role in many downstream analysis, and the lack of sequencing depth will lead to a complete failure of multi-omic studies. And also, in the study of single-cell RNA-seq, what people usually do is to filter out genes to keep only those top variable genes rather than using all the genes. And we're curious if this will have an influence on the projection. And if we focus on the heat map shown in the middle, from bottom to top, it changes from all the reads to like 1/30 of the reads. So an example is that if there are 10,000 reads in the bottom for one cell, then for this cell, there's only 300 reads at the top. And this is more severe than all the publicly available data sets. And changing from left to right, we change from using all the genes to 75%, 50%, and 25% genes based on their mean value. And we use label transfer accuracy to evaluate the success of projection. And label transfer accuracy is basically calculated by building a supervised model using the reference data set and predict the label of for corollary data set and compare the predicted and the true label. And so basically from bottom to top and from right to left, the accuracy decreases. And if we can also see this from, if we look at figure 3 comparing to figure 1, which changes from a total separation of cell types into somewhat mixture between among those cell types. And we can also see this from changing from figure 4 to figure 1. And so it means that the number of sequencing reads will be harmful to the analysis, and if only using top variable genes will be beneficial. But there is another point I can make in here, that if we focus on the setting here, the accuracy is 0.68, and is not a small number. So it represents the stability of the projection with respect to the number of sequencing reads and the number of genes we use. Overall, scGAD provides a set of analysis tools to address the pressing needs for integrating single-cell Hi-C data with other single-cell data modalities. It allows for finer cell type annotation, achieves a better cell type separation performance, and the projection with single-cell GAD is robust to change of sequencing reads. And thank Sondas and Ye for this great collaboration, and also thank all the members in our group for their great help on this presentation and this project. Our method is available in GitHub, and the paper is also available in bioinformatics. And with that, I'm happy to take any questions.
Any questions from the in-person audience? We have one here from our keynote speaker earlier, Myra Magaril. Thank you for your talk. Can you think of a way to integrate single-cell RNA-seq data with population scHi-C data sets?
Thank you. This is a great question. But I think it is possible to integrate between single-cell RNA-seq and the bulk level Hi-C data. I think the bulk Hi-C data is measured like differently from single-cell Hi-C, that we already know the cell type of that. So like after calculating the GAD value for that, we can directly like calculate the pairwise correlation between them rather than using a more complex tool to do integration. Yeah, that's what I think.
Thanks, it's really interesting. And
Thank you.
I'm curious about, I mean, I think I sort of missed when the actual how scGAD is calculated, the details of how that works. But I guess it would help me to get an intuition for, I guess you get a vector, right, in which there are entries per gene. Can you say a little bit about what the semantics of those entries are? How do you interpret the values in the scGAD?
Okay, thank you. Okay. So the question is like how to interpret this scGAD value, right?
Yeah.
So I think, are you familiar with the single-cell attack-seq integration? And in single-cell attack-seq, there are like some early method for single-cell integration between single-cell attack-seq and single-cell RNA-seq is that people will like merge the region into gene level and build a cell-by-gene matrix and like do integration using this matrix to the single-cell RNA-seq matrix. And we're having a similar spirit here. So what I mean is like biologists doesn't seem to care much about the gene activity score from single-cell attack-seq, and I don't think they will care much about this scGAD value as well. So the value itself might not represent anything, but it's like a tool to bridge between single-cell Hi-C and other assays.
Okay.
Yeah.
So I was curious, when you look at these single-cell Hi-C data sets, how much signal is there? Is most of the signal coming from genes, and how much around the gene do you look at? Because these are like global matrices, right? And the gene part of the genome is very small. So I also have a second question.
Yeah, I think I can answer the first. So the question is like the distribution of a number of the sequencing reads at on this contact maps, right? Kind of like.
Right. Yeah.
So basically.
How much information are you losing about, by just focusing on?
I think a good thing is that we're not losing a lot of information because most of the signals are actually in the diagonal and off diagonal of this of each of this matrix. So when you like at those farther distances, there are basically no count at all. So when you sum up this as a gene level, like there isn't a lot of information lost.
My second question is, so you said that integration is very important to identify cell types, and you're integrating with a single-cell RNA-seq. And you said that with just single-cell Hi-C clustering, cell type identification was hard. I was curious how much information did you gain based on integration with, compared to single-cell RNA-seq. So, with single-cell RNA-seq, you can do reasonably well with cell type identification. How much additional are you getting when you add single-cell Hi-C on top of RNA-seq?
Oh, you mean like opposite direction, whether like single-cell RNA-seq?
Right. You need single-cell Hi-C over and above single-cell RNA-seq to really get to different and meaningful better.
Thank you. And I think currently the method we're using is like treating single-cell RNA-seq as a reference data set. So like so certainly single-cell Hi-C will get much more information. And I don't think it will. I think currently it will like improve the performance of the like cell type separation for single-cell RNA-seq. But we're like developing tools to do a more sophisticated integration, so that like both of the assays can like gain something from this.